Hello everyone, my name is Manuela Raimondi and I'm a professor of bioengineering at Polytechnic University in Milano in Italy. First of all, I would like to thank the organizers of this conference for inviting me to this talk, and it's a very big honor for me. I would like to introduce myself very briefly. I am the founder of the mechanobiology group at Politecnico, where I direct two labs. I invented some frontier platforms for biological research, the MOAB, the nichoid, and the MICROATLAS that I will tell you about in this talk. My potentially competing interests are that I have patented all these inventions, and together with my students, I founded a startup company that commercializes these platforms, a company in which I hold shares. My journey in the field of cell modeling started many years ago. I was trying to engineer cartilaginous tissue in vitro by the use of cellularized scaffolds like the one visualized here in scanning electron microscopy. The engineering of a tissue in vitro is a very slow process that requires several weeks of culture before an extracellular matrix can develop and fill the scaffold pores and remodel to acquire aspects of functionality. I could analyze these three-dimensional constructs only at the end of culture, and I was desperately seeking a solution to monitor tissue development non-destructively during culture. So I had the idea to incorporate a bioreactor on a microscopy slide, and with the help of Matteo, I invented the Miniaturized Optically Accessible Bioreactor. This is where the acronym comes from. It's a high-tech microscopy slide with 3 culture chambers built on it, allowing to inspect cellularized constructs non-destructively in fluorescence at various time points of perfused culture. For example, this is how collagen secreted by cells can be visualized on live cartilage cells with a fluorescent label. So I use computational modeling to predict mass transport of nutrients and oxygen in the system in the aim to control tissue development in function of these parameters over long culture times, up to several weeks. For example, this is a computational prediction of fluid dynamics environment acting on cultured cells. So optical accessibility and control over culture parameters over long time culture was finally possible in space and time. The 4th dimension. In this period, I had the opportunity to test the platform with many very important scientists. Thanks to a collaboration with Gordana and her group at Columbia, for example, in a model of another slowly developing process, the formation of a bone metastasis by breast cancer cells. So metastasis to distant organs is the leading cause of death in many cancers. Disseminated breast cancers tumor cells leave the bloodstream through the microvasculature and colonize distant organs like the bone marrow. Endothelial cells and bone marrow stem cells mediate tumor cell proliferation in a niche called the perivascular tumor niche. So the MOB allowed to add the effect of interstitial flow to this model. We found that interstitial fluid flow upregulates endothelial sprouting, but endothelial cells self-assemble in a very loose vascular bed in static culture, while they assemble in a more dense vascular bed when perfused, likely by increased convective transport of angiogenic signals. So in this model of metastatic niche, we measured a significant increase in cancer cell survival without flow, that means in static conditions, as compared to what measured with flow, but only when cancer cells were put in co-culture with stromal cells and endothelial cells. Cell proliferated less under interstitial flow, but presented a significantly greater resistance to traditional chemotherapeutic drugs, likely because these drugs target proliferative cells. Another very slowly developing class of interactions in tissues are typical of our adaptive immune system. For example, in a lymph node, dendritic cells in green, these in green, adhere to reticular cells in yellow and in orange, and express receptors of T cells in blue here to instruct them. This process of lymph node instruction can be modeled in the MOAB by seeding stromal cells, here you see in yellow, on a scaffold, then by introducing dendritic cells suspended in perfused culture medium. Dendritic cells adhere to the fibroblast reticular cells, migrate on the scaffold, and are activated in the sense that they express a T cell receptor, ICAM1, which is represented here in red. So when antigen-specific T cells that are here represented in cyan are subsequently introduced with flow in the MOAB, they tend to adhere to the dendritic cells while crawling on the scaffold. So this lymph node instruction model can be used to test cancer immune therapies and vaccines. These are both therapeutic approaches with very slowly developing effects. And this is a very nice three-dimensional reconstruction from fluorescent microscopy. Another very slowly developing process is chronic neurodegeneration. So to test a model of neurodegeneration and relevant therapeutic, we inserted neurons with damage resembling the one induced by Alzheimer's disease in the MOAB, and we cultured these neurons for many days under conditioning of neuroprotective factors secreted by rat bone marrow stromal cells. We could measure a significantly higher viability of the conditioned neurons, suggesting a neuroprotective action exerted by bone marrow cells. So this is a 3D reconstruction of the cells populating the entire thickness of the scaffold obtained with scanning confocal microscopy. Other mechanisms of neurodegeneration can be modeled by connecting multiple MOABs representing separate compartments of a functional axis. Like, for example, the microbiota GAN-brain axis in neurodegeneration that my colleague Carmen is modeling in her very ambitious platform that connects the microbiota compartment to the gut compartment, to the immune system, to the blood-brain barrier and the model of the brain. And this model will recapitulate the effect of microbiome-secreted neurotoxins on the development of Alzheimer's disease. So no matter how complex the platform, an important limitation of the MOAB and similar systems is that the interstitial flow of culture medium does not perfuse directly the capillaries that can be engineered inside the model, in the construct, because they are not anastomosed to the perfused system. This limits the possibility to model the extravasation of such circulating cells of the immune system and their possible interactions with other cells in culture in the model. So to anastomose microvessels to a perfusion, there are two possible approaches. One, here on the left, is to engineer the microvessels in vitro and anastomose them to the circuit that perfuses the culture medium. Another possibility, this is my approach, is to implant the cell model in a living organisms, for example, a mouse, here on the right. And the advantages is that the system would be miniaturized, very minimally invasive, optically accessible in multi-photon microscopy, and integrated tissue scaffolds also would allow correction of aberrations that I will explain now briefly. So the cell model in this approach can be implanted in a living organism, revascularized, and interact with the host immune system. We recently invented this concept of the MICROATLAS, you can see here. It's a miniaturized imaging window that can be used for the intravital quantification of the host response to implantation of a biomaterial or a tissue or to administration of a drug. So it's basically a miniaturized subcutaneous window chamber for time-lapse analysis incorporating a scaffold for in vivo tissue regeneration. This scaffold also acts as a reference geometry, allowing to three-dimensionally reposition the field of view during repeated intravital observations, so in the fourth dimension again, and also correction of optical aberration, because it's very precise. We were able to visualize and quantify in a label-free manner cell invasion, neovascularization, and collagen synthesis in the scaffold. And these are the parameters up to now. And we are collaborating with Giuseppe Chirico and with the help of Claudio. So the MicroAtlas incorporate, as you can see here, 4 microscaffolds that can guide cell migration and vascular sprouting. We also incorporated position landmarks, allowing to reposition the field of view of the microscope at subsequent observation times. The animation shows how the scaffold supporting the revascularized cell model is sandwiched between 2 components, one of which is a glass cover slip. We obtained the first validation in vivo of our platform by implanting the MicroAtlas in the chorioallantoic membrane of an embryonated chicken embryo cultured ex ovo, as you can see here on the left. The circulatory system of the developing avian embryo has a number of vascular shunts found only during the embryonic stages. These vascular shunts allow systemic venous return entering the right atria to bypass the non-ventilated lungs, and they are called chorioallantoic vessels. We were able to visualize cells, macrovascular networks, and collagen in the scaffolds using label-free two-photon microscopy. We visualized effectively the microvascular network generated in the scaffold in green autofluorescence by label-free two-photon emission. We found a significant greater density of blood vessels in the scaffold compared to the unimplanted areas of the chorioallantoic membrane. We also visualized effectively the production of collagen fibers in the scaffold in blue autofluorescence by label-free two-photon emission. We found a significantly greater density of collagen fibers in the scaffolds compared to the unimplanted areas of the chorioallantoic membrane. Finally, we recently implanted the MicroAtlas subcute in mice, and this is how the device looks after explant at 7 weeks of implantation. And it's very well integrated with a moderate fibrotic reaction. We were able to visualize label-free the infiltrated cells in green here, collagen in blue, and the regenerated microvascular network in the scaffolds. And these are the tabular structures in green. So this is a two-photon Z-stack acquisition taken in vivo, and the image processing for quantification of these structures, of the biological structures inside, is currently in progress. So we were very happy with this result that showed the potential of the MicroAtlas as a very valuable tool to face major problems of reducing, refining, replacing animal testing in biodrug development, for example, gene therapies, tissue engineering, regenerative medicine techniques, nanomedicine, advanced therapeutical products, also in the field of toxicology of food, cosmetics, environment, and to test biomaterial development for implant devices. Last but not least, we would like to improve the regulation in force for animal research. Back to my original curiosity-driven question on what are the parameters influencing cell response that end up in the regeneration and remodeling of a tissue. After a lot of work on live cells, I could finally formulate an hypothesis. And my hypothesis was that the cell modifies its morphology, for example, while crawling on the scaffold in the MOAB, as I've shown. The nucleus senses such deformation through a direct mechanical link between the cytoskeleton and the nuclear pore complexes. So when the cell spreads, the nuclear envelope is strained, and this in turn activates the nuclear pore complexes that become more permeable To protein trafficking, this mechanism increases the permeability of the nuclear envelope to the nuclear import of the signals called transcription factors, and those are involved in key master switches that dictate cell function. For example, the transition of a stem cell into a differentiated cell. So I spent years trying to predict theoretically the diffusive flux of a transcription factor with the help of many colleagues, with increasing cell deformation. You can see here the output of a multi-physics computational model of mass transport and cell spreading, mapped on 1/4 of a cell. We coupled cell deformation to diffusive flow by assuming a strain-dependent diffusion coefficient at the nuclear membrane. However, to validate my hypothesis, I had to figure out how to experimentally control the morphology of cells, not only on a culture dish, but also in space and time, at the cell scale. I needed to isolate the mechanical contribution from the biomechanical one. I needed a well-defined mechanical environment that could be stable in space and time. I also needed to maintain cells in a 3D contact between each other to allow for paracrine signaling. Finally, I needed to monitor nuclear entry of signals that regulate gene expression, so-called transcription factors, again in space and time. Around 10 years ago, luckily, I met with Giulio and Roberto, who introduced me to two-photon laser polymerization. This is a microstereolithography technique in which polymerization is induced in hybrid organic-inorganic photopolymers based on silicon and zirconium, like this SZ2080. A femtosecond pulsed laser beam induces two-photon absorption in the resin, which polymerizes, as you can see here, in a volume very close to the focus of the laser. The sample can be moved in space while keeping the laser on focus, and this allows to literally write the microstructure in the resin with an astonishing resolution down to 100 nanometers. So there are many advantages of this technique. One is that these geometries are totally arbitrary. So thanks to this microfabrication technique, I invented the nichoid microscaffold. So this is conceived as a microscopic grid able to maintain cell adhesion in 3Ds in a very isotropic configuration of cells, while with maximization of cell-to-cell contact, physiologic paracrine signaling. And you see here, this is a Photoshop representation showing that adherent stem cells can retain their mechanobiological transcription factor in the cytoplasm while in the grid, and they import it in the nucleus when they crawl outside and spread on flat. So adherent stem cells migrated in the microstructure and tended to home and colonize very effectively. We had to solve some critical issues like lowering the autofluorescence of the material, which changed the photoinitiator component in the photopolymer, and finally solved this problem after a lot of years of work. So some results from this type of model, as you can see here from a three-dimensional reconstruction of cells, of mesenchymal stem cells culture in the grid. Compared to flat, we found that stem cells adhere to the internal lattice of the nichoid with reduced expression of structural proteins that form the focal adhesions, like for example beta, actin and vinculin, as shown in this very elegant co-localization study by Emanuele of my group. Also, the number and dimension of the focal adhesions were significantly lower on what's seen on these controls that were on flat culture. The three-dimensional adhesion configuration induces a significantly different nuclear shape in nichoids culture cells that become roundish compared to flat culture cells that have a very flat nuclei. With fluorescent recovery after photobleaching, the FRAP technique, we measure the recovery of transcription factor MyoD GFP, and we concluded that cell spreading was in fact a primary factor determining the permeability of the nucleus to this specific transcription factor MyoD. And this is a transcription factor involved in stem cell differentiation. We found that the TF significantly localized more in the nucleus of the cells cultured on flat. But most importantly, we were able to visualize the fluorescent recovery proportional, this is a parameter proportional to the nuclear flow, on the entire nuclei in space and time in the two conditions that were nichoid and flat. We discovered that in the nichoid, the flow is reduced and also very homogeneous as the particles flow inside from all the direction in space, as you can see here. On flat controls instead, not only the input flow is faster, but it occurs from the peripheral areas of the nucleus. And you can visualize here very well the recovery very homogeneously occurring in the nuclei of nichoid culture cells. Instead, you can see here a very peripheral dishomogeneous entry of the transcription factor from the lateral sides of the nuclei. So what happens is that flat conditions, the layer of cytoplasm in contact with the nucleus on top and on bottom of the nucleus is, this is represented in black in this drawing, is very thin. So the input flows of transcription factor occur primarily from a lateral right-shaped surface, in the sense, ring-shaped surface, in the sense that the nuclear membrane portion available for input flow of signals is partialized. We call this the effective surface in transport. The mechanical configuration able to release the tension from the cell nuclei has a very significant effect on the gene expression of cells cultured in the nichoid. So the nichoid promotes spontaneous upregulation of stemness genes, for example, leukemia inhibitory factor, the LIF, and cytokines, the LIF, and also cytokines such as interleukin 6 and colony stimulating factor 3. These are two factors that promote function and mobilization of hematopoietic stem cells. So this result is coherent with the current knowledge on the architecture of the native hematopoietic stem cell niche, in which stromal cells, in green, have an arboreal stellate morphology in 3Ds with very few thin extended extrusions that contact hematopoietic stem cells, likely regulating primary aspects of their function. Thanks to these good results, we approached large-scale fabrication of the nichoid microscaffolds. We use multifoci parallelization by use of a device that's called SLM. It's a space light modulator that basically splits the incoming single laser beam into multiple laser beams, all focused on one sample at the same time, as you can see here. We are able now to cover a surface of 1 square centimeter with 10,000 microlattices in 2 hours of continuous laser writing. So I'm very grateful to the funding bodies that rendered possible all this research that I've shown you. And I also thank you all for your very kind attention.